First, I would like to, as usual, talk about the disclaimer. The views, opinions of the talk will be mine and not necessarily representing my employer and the affiliates. So first, I would like to talk about the traditional approach that has been used in drug development, in drug discovery, which is use a lot of in vitro experiments and animal models. And that sometimes has the caveat of being costly and a lower success rate. And there has been research reporting that there's a lack of reproducibilities in these preclinical studies or discovery studies, for example. And in the past decade, we have seen a paradigm shift of the increasing use of human genetics in the drug discovery. And what that does is to look into the human data directly instead of the animals, which has the potential to increase the chance of success because you sort of bypass the detour. And that can lead to the potentially a higher success rate. And that's actually being shown through data. There's been a series of research in recent years that shows, for example, in this paper, they're using the actual data. They can confirm that drugs with the genetically supported targets are actually more successful in phase 2 and phase 3 studies. And that also lead to the higher chance of getting the drugs approved. And probably for that reason, we have seen a increasing investment in the pharma industry on the human data. And that often come as the computation collaborations. For example, maybe many of you heard the biobanks. FinGene is the biobanks in Finland. UK Biobank is a big study or consortium based on the UK population, but has been generating over the years a lot of data from genomics to proteomics and so forth for the patients in the or healthy patients, people in the real world. There's also the so-called AGD, which is the Alliance for Genomic Discovery. These are just a few of many examples of the consortium, the collaborations that generate the human data from the clinical phenotypes to the omics data and others. And the idea is that this will be potentially more beneficial to lead to the better drug development. And not just the drug discovery, we also have seen the human genetics applied in all the stages of the entire clinical development journey. For example, from preclinical to phase 1, 2, 3, regulatory approval, and post-marketing. Let's look at this one by one. In the preclinical study, the genomic data could be useful to help predict. Is that the voice okay? Yeah. Okay. To use the human data to predict the efficacy and the safety profiles of the drugs, and that can be used to prioritize the drug targets or the candidates that you want to move into the clinical. Because once you start that journey, it can involve a lot of investment. So you want to be smart in the very beginning. And when you look at the phase 1, you can use the genetic data to do the biomarker discovery, to look at for example, protein markers that may respond to a treatment, and that could be useful for your later development of clinical trials. When you get to phase 2, for example, the genetic data can be useful to optimize your dose, can select the dose that will be moved on to the later stage of clinical trials. And when it comes to phase 3, you can use the genomic data to help with the enrichment of patients. That has something to do with the clinical trial design, which I will also talk a little bit about that later. And you can also refine your endpoints. Sometimes the endpoints can be a biomarker, not necessarily will be at the primary, but it can be the secondary or exploratory. And also, and very importantly, the genomic data can be useful to assess the heterogeneity in treatment effect, which will be very important as we look into the deliver the promise of precision medicine. And even after the drug is approved, we can utilize the omics data to look at the drug safety and also the drug repurposing. So after you develop a drug, got it approved, launched in the market, you want to think about what is next. What is the next indication that you could further develop, and that will be the question for the drug repurposing. And a bit more about the precision medicine is as we know, is for the to develop the right drug for the right patients. And this often involves the particular enrichment strategies. So what are the patients you want to enrich in the clinical trials? Using this FDA guidance that published a few years ago as introduction, it was great to list a few major types of strategies for the enrichment. The first one is to decrease the variability in patients. In this case, you want to have a good, well-designed inclusion-exclusion criteria. So what the patients you want to include and exclude in your trial. And you also may want to have a better diagnostic tools. Often these tools can be based on, instead of the subjective measures, you can look at the biomarkers, the omics. I think the previous speakers also provided several useful examples. The second big category is the prognostic enrichment. Or in other words, you can think of to look at the high-risk patients. This actually is for a reason, or many reasons. One of the reasons is when you have the high-risk patients, you actually may potentially need a smaller sample size for your trials, which will make your trials more efficient. And to identify these high-risk patients, you can look at this multimodality data and coupled with the predictive analytics, as it provides brings the opportunities to leverage machine learning, AI, and so forth to predict what the patients are likely to be high risk, or classify them in these groups, which can be used to enrich your clinical trials. The last but not the least is the predictive enrichment. The idea is to identify the patients are more likely to respond or respond better to the treatment. And that's kind of actually the holy grail in clinical development. You want to know what the patients will be responding to your drugs, right? And that is the predictive enrichment. And in this case, we are looking at the so-called heterogeneous treatment effect. And what is it? Heterogeneous treatment effect is actually pretty simple. Is the situation where the response to the treatment or intervention will differ among individuals or subgroups of patients. And let's look at these figures. First, on the top, the y-axis is the treatment effect, and the x-axis is a patient characteristic. So if you look at this, it's a fairly random, evenly distributed. What that means is then if you look at these vertical red lines or in a narrow region, you can see if you calculate the average treatment effect, they're pretty similar. They're the same. So in that case, we can say that treatment effect is homogeneous. But if you move on to the lower panel, where you can see there is some trend, looks like the treatment effect is a function of the patient's characteristic. What that means is if you look at this wider range of this patient characteristic, you may see the average treatment effect, but when you narrow down to a certain range of the subgroups, you will see the different treatment effect. And sometimes that every treatment effect would lead to strong enough evidence to approve that drug, but for that subpopulation. And that's kind of the basis of a precision medicine. And there's some caveats or challenges here. First, we do not observe the treatment effect directly. We observe the outcomes of patients, and also the patient outcomes for those patients. They are either receiving the drug or not receiving that, so we cannot observe them at the same time. Another part is these patient characteristics. They can be many different things. They can be age, can be sex, can be biomarkers. And the biomarkers can be a single biomarker or a large number of biomarkers, depends on the hypothesis, the scientific hypothesis that we have available. So we sometimes we don't know what the patient characteristics matter, and that may involve some of this exploratory analysis. And I would like to talk about the pharmacogenomics in particular as a tool to assess this heterogeneous treatment effect. And if you recall that we have this axis of Y being the treatment effect and the X being the patient characteristic, when it comes to the genomics or genetics, there are different ways to measure this patient characteristic. First, it can be a genotype of a single nucleotide polymorphism. For those who are not familiar with this term, you can imagine across the human genome, there is one location that people differ just by different categories. For example, AA, AB, BB, and we call them genotype. And you can use this as a characteristic to see, to measure the genetic variations. But you can also look at it at the gene level. We have tens of thousands genes on the human genome, and there are different ways to categorize that. But here, one particular way to look at the variation at the gene level is so-called the star allele. The star alleles is specific in measuring the variation of a metabolizing status among people. Another one, maybe it's more general, is a score that will be calculated for a patient or for a subject based on a function of many genetic variants. So this can be at a gene level, it can be across multiple genes, and sometimes even across the whole genome. So what that gives us is sometimes you may have this score that is a categorical, in this case, for example, 3 categories, or it can be a continuous score. And those are the different ways to show the patient characteristic, which will then be used in this figure, for example, to assess the heterogeneity in the treatment effect. And I would like to use the rest of the presentation to focus on one particular one case study of a pharmacogenomics. I think we talk a lot about AI nowadays, and one important part is going back to the processes, the workflows, to understand what we have been doing every day and what are the opportunities for AI applications. And I actually will welcome everyone to think of this together with me, because this is ongoing research, and I will talk about the workflow. So the focus is on the workflow and not a lot of necessary the AI applications, but that could be the potential in the future research. So in this case study, our objective is to assess the heterogeneity in treatment effect using the genetic information. And that is what we call pharmacogenomics, pharma and genomics, PGX. And the input data, if you can see, look at the figure, the workflow on the right. We take the data as input from clinical trials, and we also generate the genomic data using the samples collected in those trials. And this can be a single study or multiple studies. We often consider phase 3 because that gives us a larger sample size. But keep a note on that part. And I would like to talk about some of the considerations, namely the proper uses of patient data, the data management and the access, data quality and representation, statistical modeling and inference, computations and the workflows, and the last, interdisciplinary research and teams. So in the next few slides, I will walk you through this one by one. First, let's look at the data, where the data flows in. We have the patient samples collected in clinical trials, but it's very important to protect the patient's rights. So how do we know that these patients allowed us to do the research that we want to do? And this often is captured using the forms, so-called informed consent forms, ICFs. And that can be, especially if you have not been doing these digital transformations, they're still often paper-based, and especially if you have a legacy studies, those are at the paper-based, or even if it's electronic, they're unstructured. So going through those forms manually can be very time-consuming, especially if you think of, for example, in large pharma, you have a study that has hundreds of patients or thousands across different studies that can take a lot of time. But it's very important. It's ensure that you do not misuse the data from your patients. So one of the example here, actually, this is the use of Gen AI. One pharma companies develop this framework to automate the extraction and reviewing of these forms. And our team has been, they have had the opportunities to collaborate with them and contribute to this work. This work was presented last year. I think it also got an award from this FDA competition. The idea is, I think nowadays is probably a lot of people, especially in the AI practitioners, been using the RevX to look at these ICF forms and try to automate that. As a result, you're able to cut the time of reviewing a certain amount of ICF forms from months to weeks, which is or actually hours, which is very impressive. And we'll not go into the details, but we also have been pretty careful in including the human reviews in this whole process to make sure that these decisions are well made. The next one is about the data management and access. Now you have the genomic data, you have the clinical data, you put them together. As we know, the genomic data can take a lot of space, from the raw data to the intermediate files. Depends on what types you are looking at. It can be several hundreds of megabytes, but up to at a terabytes level. And those data actually are needed for the different purposes of the downstream analysis. So you cannot really start from the smaller data. You often need those upstream or intermediate files as well. And keep in mind that we are starting from these clinical trials. So a lot of the systems built for clinical trials were not able to or not flexible in the scaling up. So our solution was to adopt a scalable system. And because we often need the different objectives, some of them could be leading to getting your findings and submit to the regulatory agencies. So we would like to have these GSP options available. So in the end, we are having this system that is both scalable, leveraging the cloud storage, and also the GSP compliant. And you probably can guess this is a Domino. We are using the SE for this. And that also provides, of course, the proper access controls for the people accessing the data and the results. Now, as we move on to the quality control part, I think on the agenda you will have a section specific on that. So I will not talk about the standard QC checks for this type of data, but specifically when you have the clinical and the genomic data, in this setting, because the data coming from the clinical trials, we would it's fair to assume that the data on the clinical side is fairly clean and ready. But when you look at the genomic data, uh, often there's a bioinformatics workflows that make the data fairly ready. But when you combine them, there may be the surprises you want to ensure. For example, in our experience, and for those who are familiar with the genomics, you could look at the genomic data to infer the relatedness of patients. And sometimes you do not expect those patients in the clinical trials are highly related, but that could be the case when you look at the data in the genetic level. And sometimes this is we're talking about biology. So there could be the genetic data that could be inferred the biological sex. And that often is important for us to understand the biology and the drug development. Another part is as we are looking at the clinical trials, especially randomized trials, there is a balance between the the treated group or the placebo or the experiment versus the standard of care. And that balance is important. And that is the foundation of the causality that we can infer when you look at the treatment effect. However, when you look at the genetic data, that may not always be the case. So important to check this balance, make sure that your studies are balanced in this sense. Or in general, you want your data to be representative of the population you are looking at. And another special consideration is not always we are able to sequence all the patients in your studies. And you may actually, by design, to get the subgroup. So in that case, you may have the challenge that you have a subset and how you are able to generalize your conclusions. The next one is about let me check the time, the statistical modeling. So now we have the data and we want to do the analysis. We going back to our objectives. What is the potential genetic basis underlying heterogeneity of a treatment effects? And because we look at these genetics, often there are a large number of markers to look at, and there's also limitation in sample size. When we look at a typical phase 3 study, the sample size can be at 100 level. Sometimes you may have at 1,000 level, but that's not often the case. So that it put a challenge on how you can look at your data to make proper inferences. And for the solutions, there are two big types of analysis, especially in the pharma, in the clinical development. We look at association testing to understand if something is a risk factor or a factor that are of interest. So in this case, you want to look at if the genetic markers is moderating the treatment effect. And also predictive modeling. Sometimes you can use machine learning, you can use more complex statistical inferences. And one of the challenges is that you do not have or often you don't have enough data. And we had an opportunity to work on a study that has thousands of patients in one phase 3, but that is probably the largest we have seen in our experience in the oncology. But even our collaborators said that was once a lifetime opportunity. So what we often consider is to leverage the large data sets from the public domain, from the real world, to inform our research. So, for example, you can previously we mentioned the different data sets, UK Biobank, and so forth. You can leverage, you can develop those scores there to take carry over to your clinical trials. Basically, you train your models using other data and you apply that in the clinical trials. But there's also inherent caveats because in those studies or in these real world data, they do not have your treatment there. So you cannot easily directly predict what you're really looking for, which is what is the heterogeneity of your treatment, of the drug of your interest. But nevertheless, there's still different opportunities to inform the research, and I have one example to talk about this utility, but there are also ways to leverage the information or borrow the information from the real world, to, even though it's not about the treatment, but that part can help with your statistical inference. The next one is the computations and the workflows. There's a lot of, because of the data, there's a lot of IOs at times. And because we're talking about the genomics, it can be millions of genetic variants and can be tens of thousands genes. And that often requires a lot of CPU times. And um, obviously, this is not feasible or ideal for individual workstations. But even in the traditional cloud-based systems, for clinical trials, they're not built for this scale up. So in this, to solve the problem, we scaled up a computing environment in Domino so that allow us to leverage the parallel computing or parallel jobs when there is a parallel jobs available. Sometimes analysis is a sequential, so that will not be really possible. And we also use the different automations, but when future direction is to leverage the features in Domino or system like Domino for the further automations. The last but not least is the interdisciplinary nature of this work. You probably have heard that I talk about these words: statistics, AI, machine learning, clinical trials, and data standards, genomics, molecular biology, high performance computing, and when we look at these the drugs, obviously there's also the clinical pharmacology involved. So for the I think especially when we look at the clinical development, the genomics, the PGS, all these are important, and you really need to integrate all of them. So a solution is to have a collaborative and interdisciplinary team for that. And you also, it will be great to have a flexible data science platform that is fit for this purpose. I want to briefly mention this example that we had with a collaborator in pharma companies. So the idea was to develop a score that can can measure or predict the severity of the disease. You apply it to this phase 3 study that has been completed, and you were able to look at the scores. And what they found was they actually were indeed predict that can be potentially predict the major cardiovascular event. And this shows the potential of using genomics for the enrichment. Here is a prognostic enrichment. So how do we find high-risk patients, which we mentioned earlier in the strategies? And here I mean mainly talk about the clinical trials, but there are also opportunities to leverage the real-world data. And sometimes in clinical development, this is actually important because when you have a rare disease or severe disease, often it's not ethical or feasible to have a control arm. And even you have, say, what about the standard of care? And for some of these unmet needs, you may not even have a reasonable set of care to use as a control group. So in that case, there's a need to leverage the opportunities in real world data. And that also has been seen in recent years that the regulatory agencies are more open to that. One difference between the real world and the clinical trials are the lack of a randomization. So how do we do that? I'll not get into the details, but combining statistics, machine learning, and causal inference, there are ways to address this. And not just use this cartoon to illustrate that high-level idea. It's so-called the potential outcome framework. It's actually pretty simple. These solid figures are the actual patients, either receiving the experimental treatment or the comparators. And imagine the same patient receive the other treatment. And if you have that, you will able to look at the differences and you can calculate or measure the treatment effect. However, you do not observe these gray figures. But this would lead to the questions, how can you predict that? And that brings the opportunities of machine learning. Of course, there are a lot of more considerations, methodologies considered, but that is the rough idea. So hopefully, through this quick walkthrough, I can give you an idea that human genetics supports the drug development and the regulatory approvals. The pharmacogenomics helps identify the treatment variations and can guide the treatment, the trial enrichment. And as an outlook, we see that the real world data has the potential to complement the clinical trial data, but obviously requires special considerations. And lastly, I think also previous speakers talk about the multi-omics. Here, we just mainly talk about genomics, but there's also opportunities to leverage these different dimensions. Yeah, and that's all what I had to share with you all, and welcome any questions. Thank you.